Now we will study some of the protection tests that if we produce some kind of vaccine or some diagnostic procedures, that how much that we have the protection from this. Especially it is important from the vaccines point of view. If we have produced some special kind of the vaccines, we need to use some protection test whether the animals or the human beings or the organisms which are already being protected, they will be given the vaccines, and how much the vaccine is the antigen, it will act as a virus, or the virus it will act as a vaccine. So for this purpose we need to use the protection tests. Some of the time the protection test may not be wholly serological. We have to use some other test to see whether the animal body or the human body is protected or not. Sometimes we are using the antigens, the viruses as an antigen. So for the identification of those antigens of those viruses, there is need to do the protection test. These is the applications of the protection test, or we can see the introduction of the protection test. It is used to produce the successful vaccines, because when we are producing the vaccines, we need to do the protection test with whether these vaccines or these viruses or these antigens are good for the healthy organisms or healthy animals also. Sometimes we are using the vaccines for the human beings, but we need to use the protection test after the clinical trials. Clinical trials are of three types: clinical trials 1, 2, 3, and then CDC has special procedure for the production of the vaccines. Before using them in the human beings, we need to see their protection test in the cell lines, in the cell culture, or maybe sometime in the live animal body. It is also employed for testing of the commercial serums and vaccine products. Then the production of either active or passive immunity in an animal, followed by a challenge dose of the virulent virus. Of course, for the protection test, we need to use the same virulent virus which we are using to produce for the serum, or which we are using to produce for the vaccines. So we need to see that how much active or passive immunity is produced by that special antigen. It is also called in vivo neutralization test, because we are using it in vivo and we are going to neutralize the virus or the serum or the antibodies. Therefore it is called in vivo neutralization test or the protection tests. Vaccine prepared for all types of immunity to different lab animals. We can use different lab animals first, and then we can use the similar animals for which the vaccines is going to being produced. Satisfactory immunity is produced, all are challenged with the known virus. When we know that what kind of the immunity will be produced after that vaccine, we need to use it for the known virus. Examples are used commercially for rabies and hog cholera vaccines. First of all, the rabies vaccine, when we have produced a vaccine, or maybe we can use the chicken Newcastle disease or the chicken coccidiosis. First we will produce the vaccines in the laboratories, and then we will do the animal trials on the smaller basis, and then on the large production basis of the vaccine, and we will see the safety results and its protection results, whether they are protected or not. Because from the safety point of view, it is very important that we have to use the safest vaccines, because safety is the first trial for the vaccines or the antigens, and safety is the first benefit of the one, and then used by injecting the graded doses. So for the vaccines we need to use some different graded doses, which is already given with the dilution that which should be the most protected dose for the preparation of that specific kind of the vaccines or the specific kind of the serums which are going to use. And for this purpose there are different kind of the protection tests which are being used.